Right. Hello everybody. Hello everybody, and welcome to the first lecture in Brain, Mind and Education. Just to make things a little bit more difficult for myself, I've decided to stream this live, okay? And this has meant an awful lot of preparation. I'll go into that in a second. But actually, the reason why you have to put up with me experimenting with this technology is because I hope in the future that this means you'll be able to access the lecture from wherever you are, and you won't have to be able to come in, you know, and you can ask me questions, and you'll be able to, even this lecture, you'll be able to look at on YouTube afterwards, if it goes okay, all right. And I guess the other thing is, please be a little bit patient with me, because what I didn't realize is that when you, I didn't anticipate, is that when you broadcast or your lecture live, you have to make sure that all the graphics materials, all the images are not infringing copyright. So I've had to replace almost all of the images on these slides, mostly with Wikipedia images that have Creative Commons licenses. Although it's been a very interesting experience, because I found out that many of the images on Wikipedia are actually better than the ones that I was using. So there is some advantage there. Okay, so my name is Paul Howard-Jones. I'm going to be teaching you over the next term all about brain, mind and education. I won't go into the aims too much of the unit. I think you understand that this is about giving you the mental tools to be able to access literature in cognitive neuroscience and make meaningful links to issues which are important to you regarding learning. There's a few timetable issues. Well, there's one in particular, which I'm sorry to have to break this to you, but next week I have to be in London on Wednesday all day. So what I'm planning to do is to do the lecture, instead of on Wednesday, it's going to be first thing on Thursday morning, 9 o'clock. Okay, it's painful for me too. It hasn't been confirmed yet because we need to book the room, but this lecture is probably going to be at 9 o'clock on Thursday morning next week, okay, not on the Wednesday. The reasons for that I can't actually express because they're embargoed at the moment. I have to go down to a press conference, but it's some exciting news about research funding for the university in terms of neuroscience and education. So it's good really. I'm afraid it's one of the pains of having lecturers who are very research active. It means that every now and then we have to sort of disappear and you don't know where we are, and next Wednesday is going to be one of those, and I'm sorry about that. The good side of that is it means that we are able to talk to you about what is going on now and what may be going on in the future. So there's two books that I would recommend for this course. Of course, I have to recommend my own book, Introducing Neuroeducational Research, not just so that I can feed the children, but also because it'll help you make meaningful links between the neuroscience and education and help you think about some of the issues that are involved with that. The other book is Jamie Ward's Student's Guide to Cognitive Neuroscience, a really excellent and accessible introduction to all sorts of areas which are relevant. In fact, I think 90% of the material in terms of the topics, you know, attention, memory, emotion, are relevant to you. So that's definitely a good book to look at. Okay, so we're going to be covering today neurons and neuroanatomy. We'll be looking at other cells other than just neurons very briefly, because neurons are our real interest. How communication occurs within them, action potentials. We will be looking at key orientation terms in terms of how to find your way around the brain. That's why you have a big plastic brain in front of you on your tables. You will be having a fiddle with that later on. We'll also be thinking about what we call the different parts of the brain. We'll be looking at different ways in which we notate the different parts of the brain. Sometimes people will say a frontal lobe, sometimes people will say Brodmann's area number 9, and what's the actual connection between those two things? And we will begin to think about locating function. Which part of the brain is associated with what sort of function, and how to think about that and how not to think about it as well. Okay, so introduce to you the most important cell that you're going to come across on this unit, which is the neuron. There are certain things you need to know about the neuron. Oh, and perhaps I should say, just remind you, that I think it's on the 23rd of October, is it? You should know that date already. Is it the 23rd? I think it is. No, it's the 24th. 22nd, thank you very much. 22nd, you see, I knew that you'd know the date. 22nd, there is going to be a short examination, a multiple choice exam on the material in this lecture and on the material in the next lecture. But both of those lectures, I'm repeating myself a little bit because some of you will have had this on induction week and some of you will have not had it last week. But I need to point out that that exam is only on this lecture and the next lecture. So if there's anything in the exam which couldn't be answered using the slides from this lecture and next lecture, then you can take me to court on that, okay, and complain about it. And you might find there is, because I've had to redig all the slides. But, okay, so this is the nucleus of the cell here. A lot of important stuff happens there. We'll be talking in a minute about how action potentials are generated that go down the axon to the presynaptic terminals. So this axon here can be very long, you know, it can stretch all the way down your leg. So this can be a very long part of the communication. And at the other end of the axon, we have presynaptic terminals here, and those are going to connect on quite often to other neurons, onto the dendrites of another neuron. So it's a little bit like a Lego brick in that sense. You know, you've got an end which is going to clip on to the front bit of another brick, if you like, or another neuron. Very important myelin sheath here, which is about insulating. It's about insulating the action potential, which is actually in the form of electricity. So it's actually an electric charge that flows down here. So when this neuron communicates with another neuron over here, an electron charge is produced, goes down there, onto the presynaptic terminals, onto the dendrites of another neuron. Presynaptic, if you just translate that, pre means before, synaptic is another, or synapse. Synapse is another word for connection. Okay, you will find throughout this unit that there are all sorts of really complicated sounding words that actually have a very simple meaning. I don't know if this stems back to a time when people like to really confuse people and make themselves sound more sophisticated than they were with their neuroscience. But even worse than that, there's often 3 or 4 really complicated words for something which is actually quite simple and can be expressed monosyllabically. Okay, so we need to talk about how that electric charge is actually generated. And to understand that, we need to think about what is happening at rest when there is no charge traveling down that axon. Well, if we have a look inside the cell, so we're looking at the purple bit here. If we look at inside the cell, this is like a diagram, if you like, a topological diagram of what we're seeing here. And inside the cell, we have got protein ions, we've got potassium ions, we've got sodium ions. Outside the cell, we have chlorine ions, sodium ions. But things at rest are designed in a particular way. The membrane of the neuron has selective ion channels that allow these ions to pass backwards and forwards under certain conditions. But at rest, potassium ions, those positive ions, cross the membrane quite easily. The chloride ions, negative ions, sodium ions, and also positive, they have more difficulty crossing. And we also have these negatively charged protein molecules inside the neuron that cannot cross. On top of that, we have a pump, which is pumping out 3 sodium ions for every 2 potassium ions that it allows in. That's a real imbalance. And what that results in is actually having a negative charge, a slight negative charge within the cell. So we have what we call a resting potential, a negative charge inside the cell of 70 millivolts or minus 70 millivolts. Now that's important because what happens as a result of some sort of stimulation, and this stimulation can often arise, for example, from a peripheral neuron in your eye receiving light. Peripheral neurons are very special in that sense. They're like transducers, taking energy from the outside, turning them into something like an action potential. But whatever it is that arrives on the dendrites of that neuron actually has a slight sort of positive charge to it, and it begins to lift. So a little bit of positive charge lifts the potential inside the cell just a little bit. And when that starts to happen, it starts to depolarize, the sodium channels open, and because the sodium channels are opening, that is actually allowing more of these sodiums in, making it a little bit more positive, and it gets more and more and more positive, like this, up to about 40 millivolts, and now there is a positive potential difference between the inside of the cell and the outside of the cell. It doesn't carry on forever because at some point the sodium channels decide to close, the potassium channels open, potassium channels allow the potassium positive ions to go out, and it repolarizes down like this and goes back to zero. And in fact, it goes beyond back to zero and beyond even the negative resting potentials. There's a bit of an undershoot here before it actually goes back to minus 70 millivolts, the resting potential of a neuron. Okay, so to give you some idea, we have this wonderful graphics, one of the many wonderful things I discovered on Wikipedia over the last few days, which I didn't have access to before. So I can show you here really there's this depolarization, so that the negative inside the cell actually becomes slightly positive momentarily, allows the action potential to move down the axon. And remember, this axon can be very, very long, you know. So this is great because actually now what you've got is a piece of electricity moving down a wire, if you like. It's the same sort of thing going on. So this is, I hope you can see that this is the previous diagram really. This is the cell body, these are the dendrites, these are the presynaptic terminals. So when that gets there, that bit of positive electricity, these presynaptic terminals are probably going to go on to some dendrites of another neuron, and it's going to carry on. Okay, and the whole thing's going to happen again in another neuron. And that means that we can have networks of neurons such as this, and some sort of peripheral neuron here, let's say, you know, light is shining on these visual peripheral neurons here, are going to cause an action potential, which is going to go down there onto the dendrites of another neuron, onto the dendrites of more neurons like this. And you end up with a network which is actually communicating changes in charge, yeah, through changes in charge, electrical charge. So information essentially is passing through this network in the form of electricity. Now that's important because it means that along the way, whatever came in here, depending on the properties of this network, can actually change and come out here slightly different. So, you know, two signals here can go in, and let's say this network is designed in such a way as to produce no output, or let's say it produces an output on all of those neurons, but only when it has an input on both of these. Okay, let's say it's a neural network that wants to be sure that it has input on both of these neurons before it outputs an answer, which says, yes, I have input on both of my neurons. Now that's actually quite an intelligent thing to do. That could be useful. You can imagine that in all sorts of circumstances. If we can change the properties of this network so that these electrical charges are distributed through it and travel through it in that particular way, then we have a network that can actually transform information from one type into another type. Here, this is just about whether light is falling on two neurons. Here, the message is about whether, in fact, light is falling on both neurons or not. One is not enough. Some sort of decision has been made. Okay, so this is actually the basis of how neural networks, our brain, thinks, if you like. Okay, thinking is all about taking in information in one form, putting it out in another form, or at least that's the way that the neural network people like to think about it. So neurons generate and mediate electrical signals in a complex and interconnected manner. And this is a very simple neural network consisting of 1, 2, 3, 4, 5, 6. Does anybody know how many neurons, we have 6 here, how many neurons you have in your brain, roughly? 1 billion? 10 billion? 100 billion? Any advances on 100 billion? Okay, the answer actually, the usual answer you hear is 100 billion. Okay, that's the thing that you'll find on the internet. Now, in the last 10 years, it has actually been measured by using a method which essentially grinds up brains into soup, takes a sample, and then extrapolates back. I think it's called fractional. No, I'm not going to tell you, actually, especially if I'm being filmed. But thanks to this technique, we know there are around 86 billion. Okay, so 100 was pretty close, but there's 86 billion neurons. And actually, even a network like this with 6 can still do a certain amount of information processing. So you can just imagine what the potential of the human brain is. And can I just say that the brain is active all the time? All the brain is active all the time, unlike you may have heard in a recent film, okay, where it's emphasized that only 10% of the brain is active. What would it be like if we used all the brain all the time? Well, we are using all the brain all the time. I have to be careful not to name the film now. I'm being broadcast publicly, don't I? Okay, so information superhighways, axons and myelin. Now, we've already talked about axons. A couple of phrases you may hear are afferent and efferent. Afferent axons carry information to a particular place in the central nervous system. So, for example, afferent axons could be carrying information about touch into your brain, to your somatosensory cortex, the thalamus. Efferent neurons actually carry information away from the central nervous system. Okay, so that could be information away from your motor cortex, eventually ending up making your leg move. Myelin insulates, reducing losses, speeding up transmission from one node of Ranvier to the next, okay? So a little bit of slowing down there, and then it speeds up, and these bits between the myelin are called nodes of Ranvier. Loss of myelin is often associated with diseases such as multiple sclerosis, which involves muscle weakness and seizures. And in fact, you can lose myelin through viral infections as well. Myelin is whitish, and you've probably heard the phrase white matter and grey matter. Yeah, well, white matter is essentially white because of the myelin, and where there is more neurons rather than axons with lots of myelin wrapped around, where there's dense packets of neurons, we call that grey matter, because in fact it is more grey. Okay, so that's where the phrase grey matter and white matter comes about. White matter, of course, is really important because it's all about transmitting the information, but we don't usually associate it with processing. So when we're imaging, if we were imaging your brain, and some of you probably will have your brain imaged this year because we'll want to know how it's working, well, some of the first things we do is take out all the white matter, because we say, oh, well, that's just the wiring. We want to know what the real components, what the neurons are actually doing. There are cells other than neurons, and cells such as glial cells actually for a long time were considered to be sort of chiefly about structure. We understand now that some of these cells are very important. For example, astrocytes here remove the excess potassium ions. You're probably wondering what happens to all those potassium ions that leave the cell. Oligodendrocytes make myelin, and they can actually, this is an oligodendrocyte here, and it can actually wrap around several axons at once. So it can wrap around this one here, and it's supposed to be showing it wrapping around that one over there as well. Schwann cells make myelin too, but they wrap around single axons, and they're chiefly associated with the peripheral nervous system. I'm using these things like the central nervous system, the peripheral nervous system. What do I mean by that? The central nervous system is essentially the brain and the spinal cord. And all the rest, where all your axons are traveling all over the place, that's the peripheral nervous system. We also have microglia. Those ones were macroglia, the bigger ones. The microglia can be very important for cleaning up debris in the central nervous system after damage, if you've suffered some trauma in the brain, for example. Okay, so just to explain again, because I've already said this just now, peripheral nervous system, central nervous system. Peripheral nervous system can be divided into two main sections. The autonomic, which is all about controlling internal organs, your heart rate, your breathing, etc. The somatic peripheral nervous system, which is about connecting to motor nerve fibers and skeletal muscle and making your body move. The autonomic system can be divided further into the sympathetic system and the parasympathetic system. Sympathetic system is very much about increasing energy use and fight or flight responses, those sorts of things, very much engaging the sympathetic nervous system. Whereas the parasympathetic nervous system is all about what happens when you're relaxing, digesting, having a bit of a rest, essentially. But the bit, of course, that we are most interested in, because we are interested in learning and education, is the central nervous system, which, and of that, the brain, rather than the spiral cord, may be what interests us most. Okay, so now I said that there were many words that you'll come across in neuroscience which appear almost there to confuse you. And I'm going to try to set you on the straight and narrow now so that you are not confused. These are some of the terms that you're going to come across. It is important to be aware that these terms in the brain are like this, but actually if you look at the anatomy, and I actually know we do have some horse lovers here, and maybe they know something about horse anatomy, but if you go and look at a horse anatomy book, actually you may find that these are in a different place. Why is that? Well, it's essentially because in animal anatomy, they were designed on the basis that the animal was down like that, and then of course the human had to stand up, didn't it? And that means that the brain, the top of the brain, is not necessarily where it would be on an animal. So be aware that these terms, in terms of this being the top and this being the bottom, apply to the human brain, not necessarily to other animals or other parts of the anatomy. Okay, so to start with, at the back of the brain, we sometimes talk about anything which is caudal or posterior. So, you know, we might be looking at something here and might say, but look, there's an activity slightly caudal to that region. In other words, we're moving in that direction. So this is not a place, it's a direction. Okay, it's not a place, it's a direction. Helps you find the place. So we might talk about a caudal region of the frontal lobe. In other words, not this bit here, but that bit here. By the way, just to explain, this is the front of the brain. We know it's the front of the brain because you can see this cerebellum here sticking out the back. I mean, I forget, of course, so you're not used to looking at a brain, and it's quite important. By the end of this unit, you'll look at this and you'll just know that that's the front. But probably I need to emphasize that now. This is the front of the brain, the frontal lobes. So when we start going to the back of the brain, we're going caudal or posterior. Both these words mean pretty much the same. How confusing is that? Yeah, just there to trip you up. The opposite of that is rostral or anterior. Okay, so supposing we were looking at this bit here and we said, oh yes, but a little bit further forward, but we don't say forward, we say a little bit more rostral, a little bit more anterior. Okay, that's the way that people speak about these things. Similarly with up and down, that would be much too simple, wouldn't it? So we say dorsal, I always think of the dorsal fin, it always helps me remind me about that. Superior, that's fairly obvious. Dorsal or superior, superior is on top, isn't it? I think you can remember that. Ventral is the opposite of dorsal, and inferior means down there, okay? So you might want to say, well, it's in a region a little bit more ventral caudal, you know, so you're probably going down there a bit, okay? So soon you'll be able to amaze your friends with all this verbiage. Okay. There's more of these terms, and they are important because you're going to come across them in the papers, and I don't want them to faze you. I'm going to be using them in the lectures as well. Okay, so sagittal plane goes through like that. Okay, sagittal plane. The coronal plane goes through like this. Okay, like big ears or something, coronal. How you can remember these things. There's probably an easy way to remember them. You can probably think up one and send it to me. And then the other one is transverse, like that. So if I show you a transverse plane of the brain, it's like basically cutting off the top of my head like that and looking inside. We will see some of those later. Also worth remembering, which I don't seem to put on here, okay, is that is also often referred to as medial when it's right in the middle, okay? So the medial plane of the brain is like that as well. It's a little bit similar to sagittal. If something is on both sides, so let's say, we've got activation here, and we've also got activation here, then we might say there is activation ipsilateral and slightly inferior. Okay, you begin to get the hang of it? Means just the same side, that's all ipsilateral means. When you use your right arm, you get contralateral motor cortex activation over here, right? Because it all turns around in the brain. So in fact, when you move this side of the body, it activates motor cortex on the other side of the brain. So that's contralateral. That's another word that you're going to come across. Okay, other things you might find, bilateral means both sides. We have bilateral frontal activation. It just means activation at the front of the brain on both sides. Oh, medial, I have put that on there. Lateral means away from the middle. So if I said it was medial, it would be quite close to the middle. If it's lateral, it's away from the middle. Proximal and distal. Proximal means near, distal means far. So there is an activation quite distal, means a long way away. Okay. There is an activation proximal to the ventral striatum, which means it's not actually in the ventral striatum, but it is pretty close. Okay. Now, this might look a little bit complicated, but actually it's just reversed. This part of it here is just the reverse of what I showed you before. We've got the nervous system divided into the peripheral and the central, and the peripheral is divided into the autonomic, the somatic, and the autonomic is divided into the sympathetic and the parasympathetic. We've just been through that. The central nervous system is what we're really interested in because it's got the spinal cord, and we're not really interested in that, are we? I don't think. This is really interesting, but it's not necessarily something which is absolutely vital to learning within the school context. I know some people would argue, but then we get to the brain, and the brain can be divided into three parts: forebrain, midbrain, and hindbrain. And there are other words which tend to describe these parts of the brain. So, for example, the forebrain, sometimes we talk about that as composing the telencephalon and the diencephalon. And the telencephalon is all the cerebral cortex, plus a few other parts like the basal ganglia, hippocampus, amygdala. Why do we have these? Well, I think the reason why we have these cephalon-type phrases is because really all of this is just like a big blob when you look at the developing embryo. Okay, so it's quite helpful to put all those together. But most of the time we talk about forebrain, midbrain, and hindbrain. Okay, so the brain is the forebrain, the midbrain, and the hindbrain. But one really interesting thing is the difference in proportion of the human brain in this regard to other animals. Somebody has helpfully coloured this in, so you can see that the telencephalon, which is an important part of the forebrain, here, that's in the human brain. Look at the forebrain in the shark there, which is almost sort of, it's minuscule, isn't it? So humans have a massive forebrain compared with some animals, and there are all sorts of arguments for this, mostly around evolutionary issues, because we are very social beings. All primates are very social beings, and the social complexity of our lives makes it appropriate that we should have adapted to be able to make very sophisticated, complex social decisions, and that's why we have a huge forebrain. It's not quite as simple as that. It's also bound up with our change in diet, but we haven't got time to go into that. But for the moment, just be aware of the fact that the forebrain of the human is much larger than other animals. Now, I said when you look at the embryo, and this is 6 weeks after conception, pretty much you can see that the telencephalon, you can see in the diencephalon, that it makes more sense really to divide it up into these types of category. But when you look at the adult brain here, you can see that the forebrain now is very large, and the midbrain is almost completely hidden if you're just looking at the outside of the human brain. You can't see the midbrain at all. It's very small. It's really, that's all it is. And then you've got the hindbrain down here, which of course includes the cerebellum. Interesting part of the cerebellum. We used to think it was just about balance and about movement, but it probably represents one of the sort of darker areas of the brain that we're just beginning to unravel its significance. And it's not just about movement. It has an awful lot to do with timing and cognition as well. Okay, so we ought to talk about the hindbrain briefly. In fact, I think I already have done to some extent. It starts off with the medulla, which is important for breathing and blood circulation, those autonomic responses. The pons, a little bit higher up, which is about relaying movement, regulating breathing, taste and sleep, those very basic life support issues. And then the cerebellum, which is this cauliflower at the back, which is important for motor coordination, precision and accurate timing, and much more. So, for example, the cerebellum is featured in some explanations of dyslexia because the cerebellum, if there's a problem with the cerebellum, that may introduce timing deficits, and you can't hear or distinguish in time different speech segments. So, you know, even though it's about timing, suddenly it becomes part of a very important bit of learning for schools. And then we get to the midbrain, which is tiny, tiny, tiny really compared with the rest of it, but very important because in the midbrain, this is actually, we have the tegmentum where there are nucleus. When we use the word nucleus, we're talking about a very densely packed group of neurons. Nucleus, the plural is nuclei. And it's got an awful lot to do with motor coordination. And in fact, it is dopamine from these areas which goes up into the reward system, which is very involved with decision making and our gratification. So we will return to the midbrain later on when we're talking about her motivation. And then we get to the bits we know and love, or the bits which you probably think of most when you think about the brain, which is the, okay, there are some small things, subcortical structures like the thalamus. This is the sensory gateway to the cerebral cortex. All your senses go through the thalamus, okay? And the thalamus can actually be mediated by the cortex. So if you really don't want to see something or you don't want to hear something, your cortex has some say, if you like, in being able to mediate how the thalamus is processing that information. That's a very hand-waving explanation, by the way, for the viewers at home. But there is one sense that does not go through the thalamus, but actually goes straight into the cortex. Do you know what that is? Smell, yeah, straight in. Which is probably one of the reasons, and here I am speculating, why smell can be so evocative and it's so difficult to ignore. So we have the thalamus here, and underneath the thalamus, hypo meaning under, we have the hypothalamus, which is basically about maintaining homeostasis. So if your senses via the thalamus are saying you're particularly hot, then the hypothalamus, conveniently situated to receive the instructions, will do something about changing your body state. Okay, so that's very important. And then we get to the forebrain, and there are still structures here which are not the cortex. And I'm afraid this is one diagram I could not find, so this is a Paul Howard-Jones diagram. Nevertheless, I hope that it's clear. We have structures such as the striatum, caudate and the putamen. We have the nucleus accumbens at the front of the striatum. Nucleus accumbens is used to be thought of as the pleasure zone because it activates with sex, drugs and rock and roll. We think about it more now as very important for guiding your approach motivation. So anything that you really desire, the more you desire it, the more the nucleus accumbens activates. And we also have the amygdala, and the amygdala is at the end of the hippocampus. Hippocampus, very important for encoding memory. The amygdala, very important for your emotional processing. So here are two structures which are very close together. When we get two structures that are close together, there's probably a reason for that. And actually, this facilitates emotional memory. So there's all sorts of ways in which motion mediates memory, and that is facilitated by these two structures being close together. You can see the hippocampus here. Hippocampus, very important for encoding memory. Okay? It's not necessarily where your memories are stored in the long term, but it is involved with ensuring they are stored in the long term. Okay, so that was the forebrain there. The cerebral hemispheres, let's talk about the cortex. That's the wrinkly bit of the brain. Now, you're beginning to see what you've got here actually in front of you. If you want to, feel free, please, you can sort of separate it out. And unfortunately, this brain does not come apart a great deal, but it does come apart at least in terms of the two cortices. So, yeah, we can have a cerebral hemisphere on each side of the table. And you'll notice, one of the things that you notice straight away about the human brain is how wrinkly it is. Yes, it is more wrinkly than other brains. One of the reasons for this is because, well, one of the reasons that's been put forward for this, actually, does somebody want to tell me one of the reasons why it's so wrinkly? Probably says on the back. No, it doesn't. It doesn't say on the slide. Sorry. Yeah, to fit in as much area as possible. So there was an evolutionary pressure for us to be as sophisticated as possible, and therefore it's the higher layers of the cortex which appear to be important for higher order processing. So you know, why not stuff it all in, have a big area of cortex, but crumple it up and put it inside the skull? In fact, if you open it out, it's been estimated that the original area is a double page of a tabloid newspaper. So it's about that big area. It's crumpled all in. Actually, this is being disputed now. It's being said that our brain is really not any different to that which you would expect from a primate of our size. This is very important for understanding how evolution should be involved with our thinking about cognition. We haven't got time to go into that now, but we will be talking about it at some point. And it's been said that the wrinkling process is just something that happens naturally due to the physical properties of the brain once you reach a brain of a particular size in a primate. Right. Anyway, that may or may not be the case. What we do have is two hemispheres, and they are joined together by commissures, and particularly the corpus callosum. So you will see in white, you see that patch in white, that's the corpus callosum. Maybe I should just pick one up and just point to it. But okay, so this is the corpus callosum here. You would have to have cut through that really to separate these two hemispheres out. There's an information superhighway. There's information streaming backwards and forwards between the hemispheres. We never think with just one side of the brain or the other side of the brain, unless you are one of those people who underwent radical split brain surgery in the 1950s and 60s, where they did cut the corpus callosum. Very interesting case studies. The important thing for the moment, really, in terms of finding a way about the brain, is that the ridge of one of the wrinkles, the top part is called a gyrus, the plural is gyri or gyri, and the valley is the sulcus, or the plural is sulci, yeah, or sulci. There's no right way to pronounce Latin, by the way. It's dead language, just for the viewers at home. Very important, you have 4 lobes. Okay, there's 4 lobes, and it can be a right or the left version. Frontal lobe at the front. So if you pick this up, you should be able to see these now. Frontal lobe at the front here. You've got the parietal lobe. If I get back in camera. Hello. Frontal lobe at the front here. You've got the occipital lobe at the back, the parietal lobe between. This is the temporal lobe. Okay, and you do need to really drill those into your head. They are that's really something you need to be able to have at the tip of your tongue and know where they are. There is also, however, hidden cortex. Okay, above the corpus callosum, there is, but not so far up that it's visible. You have an island of cortex, which is called the cingulate cortex. Cingulate is Latin for island. Okay, and if I was to say posterior cingulate cortex, you'd think, oh, posterior, that means at the back. So I'm talking about posterior cingulate cortex here. Okay. And there's other types of cortex. Okay, so let's have some fun. I'm sorry that the viewers at home can't do this. Apologise for that. We're only going to be 5 minutes. So 5 minutes, I would like you to just colour in and find on your brain these areas: the inferior frontal sulcus, the superior temporal gyrus, dorsolateral prefrontal cortex, superior frontal gyrus, anterior cingulate. You know where these are, okay? You have to translate the words. Yeah. You have to translate the words. Yeah, I think you can use either one, but you'll have to put it in on that one for the outside, and that one's the interior cingulate. So that's a sagittal section. Is that
not like through the middle as well?
Oh yes, it is. Well spotted. Oh, that's a shame. Yeah, something went wrong with the photocopying. I think there are some at the back. I will have a look. Yeah, there's two types of diagram here. Everybody should have had two types. No, oh yes, all right, yes. Yeah, there's two types of diagram. So you need that one, don't you? This could be forever. I could take all day doing this, couldn't I? Yeah.
It's going to be able to work next Thursday. Does that?
Would it be any good if I tried this and you watched the whole thing on YouTube?
It would be absolutely amazing.
That would be cool. Okay.
So
Yeah, I thought that might be a problem. I don't know how easy it's going to be because I've got to be in the tiered lecture theatre to do it. But actually, the whole lecture is already recorded with an audio commentary anyway. I think there's no way I can hold it against you if you can't make it, is there? Yeah,
That's it.
Okay. Yeah, there's supposed to be two. You're supposed to have two diagrams, and they were supposed to be on either side of the paper, but they're clearly not. So if you haven't got both of those, let me know, and I'll give you the other ones. Which one do you want? That one? That one. Which one is it? Oh, that one. No, you got that one. That's it. You want one of them? Oh, yeah, you got both of them there, but you might want one of your own. Is that what you're saying? Yeah. That's okay. No. Yeah, I'm sorry about this. It's because it was supposed to be double-sided. I didn't realise when I gave them, when I was putting them out. Have that one.
The other.
And yeah, you need 2 and 4.
Thank you.
I think so.
So you should have one of them. You should have one of them.
Thank you.
Yeah, this is the photocopying error. Hello, how are you? Have one of those. Have one of those. Grey ones.
Yeah.
I was so focused on the videoing technology that I didn't keep an eye on the photocopier.
Thank you.
Thank you. Well, inferior means below. Frontal is at the front, sulcus is a valley. So go to the front of the brain and look for the lowest valley.
I only see on the outside. You're not in the medial vein.
So
I'd say it was like
inferior frontal, so frontal is at the front, so it's going to be somewhere around here. Inferior means at the bottom, so and it's going to be yeah, it's a valley. So I would say it's probably that.
where is it on the, is this one? This?
Yeah. You wouldn't be blamed if it was wrong, because that is the lowest value you can see. Do you need a few clues? Because I can see that people. Okay, so look, inferior frontal sulcus. Okay, let's translate this. First of all, frontal, that's the lobe. Okay, so it's at the front of the brain. Inferior means down at the bottom, and sulcus is a valley. So you're looking for the lowest valley at the front of the brain. Superior temporal gyrus, Superior means front, temporal is the lobe, gyrus is the ridge. So you're looking at the highest ridge in the temporal lobe. Okay?
Is that relevant? All right, it's all wrong.
You're almost there. I mean, that's actually the corpus callosum, I think. So if you just moved it to there, just rub out the first bit, okay. You're almost there. Yeah, that's good. That's good. Superior frontal gyrus, excellent. DLPFC is actually going to be a band. Yeah, because that would be dorsolateral, but well, it's lateral, so it's away from the medial. It ends up being a band. Yeah, like across like that, really. Yeah. Are we doing for time? Oh, we're doing fine for time. How you doing?
I draw my own little brain. So.
Oh, very good. Yes, that's, I like it. I like it. Yeah, if you finish, I can see some people have already finished. Do feel free to find your way around the rest of the brain using this crib sheet, okay, which has got all the numbers on it relating to the brain. I think some of the numbers are on one hemisphere and not on the other. So if you can't find the number, it may be on the other hemisphere. Ooh. Hi.
Hello. The temporal lobe.
Yeah.
Does it kind of stop there?
Yeah.
That line.
Yeah.
Or that sulcus.
Yeah.
Is that a sulcus?
Well, it's a fissure. It's a fissure.
Right.
You're going to ask me which one it is now. Take the microphone off. Oh, battery's running out. Perisylvian fissure, is it? I'm not sure actually, but if you refer to it as a fissure.
And can you tell me what the difference is between a lobe and a cortex?
Yeah, the cortex is divided into 4 lobes.
Right. Okay.
Okay.
Yeah.
Okay, so I hope that you've enjoyed that little task. If you want to find out what the answers are, So this gives you some idea about whether you were close or far away, or whether you were proximal or distal. So the inferior frontal sulcus is the lowest valley traveling along the front. So it's going to be roughly about there. This is not a wonderful diagram for me to show it on, but if you've got it roughly around there, you're doing well. Of course, you could have talked about the right inferior frontal sulcus or the left inferior frontal sulcus, if you want to be more specific. The superior temporal gyrus is going to be the highest ridge traveling along the temporal lobe. Superior is higher, temporal lobe, gyrus means ridge. So you just go for the highest ridge before you get to the fissure that divides the temporal and the frontal. Dorsolateral prefrontal cortex is a little bit of a difficult one, that in some respects. Remember I said lateral meant both sides or away from the middle. So in fact you are looking bilaterally across both. And dorso is at the top. Frontal is the frontal lobe. Prefrontal means just behind, right at the front. But it's such an important area. You're going to hear this DLPFC abbreviation quite a lot when we're talking about working memory. So this is a very important region for working memory. DLPFC, it's a band running across the front of the brain here. Okay? I'll see somebody filming me, but you shouldn't have to film me because it's going to be all on YouTube, I hope. Superior frontal gyrus, highest ridge along the frontal lobe. So you go to the highest ridge on the frontal lobe, okay? Take it as high as you can. Anterior cingulate is the front of the, anterior means front, cingulate is island. So you've got to look at a medial sagittal section of the brain. You're not going to do it on the pictures that show the outside of the brain. You've got to have a slice going through like that. Anterior means front. And this is a very important part of the brain for decision making and for concentrating. So if you're concentrating on what I'm saying, your anterior cingulate is going to be activating. Okay. One day I'm going to be able to determine if your anterior cingulate is activating whilst I'm talking to you, I'm sure. Are there any questions about that? Yes, sir.
Sorry if this is a stupid question, but are all the folds in the brain identical?
It's a very good question, and no, they're not. For neuroimaging, we like to pretend that they are, so we have to morph everybody's brain together. But generally speaking, you will have the same, gyri and sulci, but actually there is some variation in that as well. So, it could be a different place. And we know also if you, for example, different people will activate different parts of the brain. So different neural networks will be involved. So yes, every single brain is unique. Every brain is different. We try to pretend when we're doing neuroimaging that they're all the same, so we morph them all together. But in reality, every brain is different and unique, and the position of the gyri and the sulci are going to be unique and different as well. Another way of... Any other questions before I carry on? By the way, I'm just aware of the fact that your question is being recorded on YouTube, so I hope that that's okay. I should have said that. I think I did say something to that effect at the beginning. Any other questions? We're all quiet now. Okay. So another way of identifying different parts of the brain is to actually look at the cytoarchitecture, the actual fine-grain architecture of the brain in terms of the graininess, in terms of the thickness, in terms of the actual appearance of the cortex. And we find that changes as we go across. Slightly different cellular architecture and composition, density of the brain. So Brodmann looked at that very closely, and he said, look, this is interesting. You can actually divide up different regions of the brain in terms of their architecture, that microarchitecture, cytoarchitecture. So he numbered all of those. What you can see here is in this rotating brain is all the different regions, and there's very many of them. And they don't tend to cross over lobes. They tend to subdivide the lobes, so they're quite useful in that sense. And some of the most popular ones, or the ones that you hear sort of quite a lot about, are primary somatosensory cortex, BA1, 2, 3, the motor cortex, BA4. And you can see already that we are beginning to identify function with these different Brodmann areas. In the papers, you'll see them referred to as BA1, BA2. They won't tell you what BA means, so you need to remember it means Brodmann area. And you can look at a map like this, and you can see exactly what part they mean. And if they say Brodmann area 38, then you know that in fact it's in a frontal region of the temporal lobes, you know, up here, and you can describe it in that sense as well. Brodmann's areas 5 and 7, secondary sensory cortex. Brodmann's area 6, supplementary motor cortex and premotor cortex, part of the brain that is involved with planning, planning your actions. 946, 9 and 46, dorsolateral prefrontal cortex. Brodmann's area 24 and 32, anterior cingulate. So instead of saying the anterior cingulate, you could say Brodmann's area 32, 33, or whatever. Primary visual cortex, Brodmann area 17, right at the back. And you know, you're beginning to get the impression that actually when you look at the microarchitecture of the brain, the cytoarchitecture, you're beginning to see there is a correlation between the region and a particular function. We have to be careful not to take that too far, of course, because even simple things are very distributed across the brain in terms of the parts of the brain that are involved. So it's not like you have a little box here for memory, a little box here for, well, actually the way they used to think about, these are some of the things that they used to think about. So they used to say, back in the old days of phrenology, which is not very long ago really, about 100 years ago or so, they used to say, oh yes, section area number 7 here, where's this area number 7? I think it's there, isn't it? That's your secretiveness part of your brain. Okay, you also have a self-esteem part, a love of approbation. I don't even know what that means actually. Wonder, ideality, firmness, hope, individuality. I think there's love of your parents is somewhere in there as well. Causality, cautiousness. Where's the cautiousness part of the brain? Number 12, somewhere there. Oh, they are, cautiousness. Okay, looks like the inferior parietal lobule, if you ask me. Cautiousness part of the brain. Okay, so this is ridiculous, obviously, but it's a very infectious idea, and it's very easy for us to get into this sense that we can divide the brain up into terms of functions that make sense to us. I mean, there's no doubt that our cognition takes place in our brain, well, most of it anyway. But you know, it doesn't mean that the way in which we think about our thought processes in an everyday sense translates to different locations in the brain. Okay, so you know, some of these words like adhesiveness, but we may think about sticking power. This is back in the days of the British Empire, and probably sticking power was probably a really important thing. You know, everybody thought, well, obviously, we know it makes sense to have a sticking power part of the brain. So we go into the brain and we look for something that is to do with sticking power. But actually, you know, the British Empire didn't design the brain. The brain is very distributed. And when you look at even a simple thing, and we'll look at a minute at a very simple thing in terms of just thinking about picking up a cup of soup, all parts of the brain are shimmering with activity all the time. So if you really wanted to see the brain, if we had the technology to do it, what you would see is shimmering activity, moving incredibly fast, distributed across the brain and changing constantly. We haven't got the technology quite yet to see that, but one day we will. There are certain parts of certain functions that are quite primary. It does make sense to think about attention, for example, emotion, perhaps memory. But you know, even memory is very distributed. So you might think about the San Francisco Bridge, for example. That's obviously an example that's often used in neuroscience books. But you've got color, you've got location, you've got associations. You know, all of those things actually are going to be activating different parts of the brain. The word San Francisco Bridge, millions different, not millions, but many different memory systems involved. So memory is distributed and many simple functions are distributed. So let's just think about something very simple. I had to find a picture of a cup of soup. It didn't have any copyright infringement involved. I found one. Here you have a cup of soup, making me hungry. It must be approaching lunchtime. And you know, all you're thinking about is you're just touching this cup of soup. Okay. And I want to give you some idea of what's involved with that, because back in the days of phrenology, it might have been just activating the cup of soup box, okay? But it's not like that. Motor and somatosensory functions are topographically organized. That's the first thing to realize. So, you know, in the motor cortex, your fingers, your hand, your arm, they're close together. And they're close together in terms of motor, in terms of output, making things move. And they're also close together in terms of the sensory information that's coming back as well, because those two things need to work together if you're to feel your hand around a cup of soup. So we have a somatosensory strip here, and we have the motor cortex there. The first thing is when you touch the cup of soup, of course, we've got sensory information. And where does sensory information come from? Where does it enter the brain? Thalamus. Thalamus, yes, you know that now, the thalamus, good. Okay, so the information comes from via the thalamus into the somatosensory strip. So that's maybe the first, let's just say that's the first thing that's activating. Okay, so red means it's on fire now. Dark red means it's going there. Okay, that's where it's traveling to. So the information from the thalamus reaches the somatosensory cortex, and from there to the motor regions, and also to the unimodal association area here, allowing the shape to be formed from touch. Now, we haven't got any other senses involved at the moment. We've only got feeling, but we've actually got quite a lot of different types of feeling information coming in. We've got feeling information from that finger, and also I can feel my hand sliding across the table in order to reach a cup of soup. This is making me hungry. All of those are separate streams of information. They need to be integrated to make up a kinesthetic, if you like, image or concept of this cup. So they need to come together and we need to know how those different streams of touching information are related to each other. So they need to come together in the unimodal association here in the parietal cortex. From there, it will feed to premotor areas preparing movement in respect of feeling the shape, okay? Because you need to know, if you're thinking about putting your hand around this cup, you need to be planning now, don't you? So, you know, your motor cortex may be activated because, in fact, you're moving your hands, but also you need to now be planning in terms of this information, how you're going to take your fingers around the cup and pick it up. So we're seeing activation coming from into the premotor cortex here. And it also goes to higher order, more posterior unimodal association areas here for putting this together into the shape of a cup. From there, the information starts entering multimodal association areas. So we need to integrate this now with a whole load of other information. Actually, I'm seeing the cup. I haven't even talked about that yet, but there's information coming in from the occipital lobe visually that needs to be integrated with this touching information. So there's activity coming in here now and joining up in these more posterior regions. Also, I can hear my croutons crackling. It's lovely. And so, you know, auditory information is coming in, and there's timing issues as well. You know, we need to think about the timing that's involved. All of this stuff is coming together in multimodal association areas. Parahippocampal regions are now activating as well. And memory, because now you begin to think about what happened the last time you picked up a cup. You're also beginning to feel really hungry. The ventral striatum is going to be activating at this point because actually you really desire this cup. There's an awful lot of things on here which are not even shown. But you can imagine that just moving your hand over to the cup of soup, activity right across the brain. So it's very important to realize that even very simple tasks activate very wide regions of the brain. And it makes it very difficult to say, oh, creativity or is really about this part of the brain or that part of the brain. You know, those are much more complex cognitive issues than simply reaching out for your cup of soup. You know, so right from the start, try to drop this idea of phrenology. Yes, we have some functions which are associated more with some brain regions than others. But when it comes to everyday tasks, those are going to recruit a very wide distributed range of brain regions. So how do we know anyway that these brain regions are activated? What techniques do we use to try to discover these things? Well, there are many, and they all have different advantages. They all have different disadvantages. I have never used, or really, I don't really know anybody that has actually used positron emission tomography. I guess one of the reasons for this is because it involves ingesting radioactivity. What happens is, whether you're eating it or are being injected with it, activity is generated by this radioactivity collecting in your brain, and then they put you inside this big scanner to pick up the radioactivity. Okay, positrons are positively charged particles that can be picked up by this sort of apparatus. Now, obviously, there's an issue here in terms of using PET for research, especially academic research. Whenever we do any sort of method, whenever we think about any method involving human participants, we have to think about ethics. Ethics are a matter of balancing up risk and benefit. I am very interested in encouraging children to learn. And encouraging children to learn is a really important thing. But it's not quite the same as curing somebody's cancer. And therefore, I have a different set of benefits to weigh up against the risks. And of course, ingesting radioactivity does bring with it some risks. And in fact, there is a limited number of PET scans that you can have in a lifetime from a health point of view. And that means that there is, you need to be even more limited when it comes to giving PET scans to people for research purposes, because you need to leave a little safety margin so that, okay, maybe they've done 2 or 3 research studies using PET, but at least we know if they need it, that they can have, half a dozen PET scans for their own health should they need it. Okay, so what you don't want to do is to exhaust somebody's limit in terms of the amount of PET scans they have, so that when they need urgent medical treatment, your research has essentially put them at risk of not being able to have it. So I guess that's one of the reasons why PET is not terribly popular in terms of cognitive neuroscience. Having said that, it's kind of, there are some things that PET does incredibly well. We're beginning to learn how to do those things with other types of scanning. But one of the things it does is, because you're sending in these chemical messengers that collect in the brain, you can actually attach your radioactivity to molecules which you have particular interest in terms of how they respond, with how they interact with the brain. So, for example, you can actually pinpoint the extent to which particular neurotransmitters are operating in different parts of the brain by making sure that your radioactivity is responding to those types of messages. Yeah. So that can be very helpful, and most other sort of methods are not quite so good at doing that. However, it does have, and I think you can see here, it has quite poor spatial resolution. I mean, if you're looking at that brain and you're trying to work out whether this is superior temporal sulcus or maybe it's just the inferior temporal sulcus, it's quite difficult to tell. So it's all quite fuzzy, isn't it? Because we know that, that actually what's going on here, well, ultimately we want to know what's happening at a neural level. We've got 86 billion neurons in here, and if all you're seeing is a big wodge of activity there, it's not really telling you very much, is it? Even in terms of Brodmann areas, it's quite difficult to identify, one particular Brodmann area. Some of these may be going over several. So the spatial resolution of PET, in terms of being able to tie down what's happening within half a centimetre, it's quite difficult. The temporal resolution, in terms of knowing when it's happening, is also quite difficult. It's not great. It's actually worse than fMRI, because you're waiting, and you're having to wait for these the radioactivity to arrive in the place that you want it to. So PET, not that great, but in terms of accurately targeting particular functions, particular parts of the brain with using chemical messengers, it's great. EEG, otherwise known as electroencephalography, I'm very disappointed actually, because I've just been told I haven't won some money to get some EEG equipment, which I'm quite disappointed about. But I tend to be more fMRI. But the reason why I'm interested in EEG, there's two big reasons for it. One is because it's got really good temporal resolution. And the other thing is that actually it's very portable as well. So it's not very invasive. They are now developing systems that you can actually walk around with, wireless systems. The way it works is to pick up changes in electrical field activity above the scope. Why would there be changes in electrical field above the scope when you're thinking about something? I mean, why would that happen? Yeah.
Because it's the change to electrical charge of the neuron.
That's right, yeah, because what you've got are electrical charges moving. And if you think back to your school physics lesson, you'll remember that when a charge, when a wire is moving, in other words, a charge is moving, then in fact you're generating an electrical field, aren't you? Okay, so that's the basis of a lot of physics. So if you're moving a wire, then you're going to be changing the electrical field, and you can pick that up. So you're basically picking up the electrical field due to the neural activity, and there's nothing mediating it. It's not like you're waiting for some blood change. You're not waiting for some radioactive messenger to arrive or anything like that. It happens straight away. So this means it's got fantastic temporal resolution. You can tell within milliseconds what's going on. It's also very uninvasive. You don't have to lie down inside a very big machine. You just have to wear a silly hair hat with lots of electrodes. So that means you can use it with children. There are also particular types of electrical signal that you can pick up that are related to particular types of behavior. One of them is negative mismatch. So if you are hearing two stimuli, as soon as they become different, your brain will produce a certain type of response that can be easily detected above the scope. When you use EEG like that, you say you are measuring event-related potentials, ERPs. It's an ERP study. An ERP study is using EEG in a particular way. If you're not using ERPs, then you're probably just translating all of the electrical activity and decoding it into particular wavelengths. The sorts of wavelengths we're looking at are things like alpha, 7 hertz to 14 hertz. A hertz is the number of cycles per second. So these waves are going up and down within this frequency range of about 7 to 14 cycles per second. That's why they're quite sort of longish, really. Not as long as theta, but longer, much longer than these beta waves down here. And these alpha waves tend to emerge with relaxation. So you get more alpha brain waves when you're feeling chilled out. Beta, on the other hand, become much more dominant when you are anxious. And beta are much higher frequency of brainwave, between 15 to 30 cycles per second. Theta, the lowest of all of them, the 4 hertz to 7 hertz, relaxed, meditative, creative states appear to be associated with theta. Now, to be creative, you need to have a certain type of alpha theta ratio. It's been found out that a certain type of alpha theta ratio is particularly beneficial for creative performance. So, for example, students at the Royal College of Music in London have found that by observing their own neurofeedback, their own brainwaves, they're able to find the zone where the alpha theta ratio is just right for them to perform well. And by training themselves doing that half an hour a day, they were able to increase the quality of their musical performance by a whole degree classification grade. Very impressive. And this has also now been tried out with school children in schools. The same professor, John Griselier, if you want to look it up, at Imperial, I think. So interesting work that's got real relationship to neuroscience in education. Now, the method that I prefer, I suppose is because I've done more of it than the other methods, is functional magnetic resonance imaging. Now, this requires putting the participant into a very strong magnetic field. And The magnetic field strength is about 20,000 times the magnetic field of the Earth. But as far as we know, there's no health risks associated with that. We don't tend to do it with women if they're pregnant, but actually there's no evidence of it affecting the embryo. And so you can have one of these as many times as you like. I've had 8 of them, and I'm perfectly normal. So here, what we do is we pulse the magnetic field, and we find out the way in which that magnetic field relaxes back to where it was before. We pulse it with a radio frequency. And the reason why that allows you to find out about function, oh, I should have said before, sorry, with the EEG, not the EEG, the PET, the important thing there is, of course, you're finding out about changes in energy, because when you think, and this part of the brain is, let's say, active more, it needs more energy, so it's more likely to absorb radioactivity if it's attached to energy providing materials such as glucose. With fMRI, you've also got a response that's related to blood issues in blood flow. Here you've got active neurons consuming more oxygen, but what happens is actually an increase in blood flow occurring without the matched oxygen extraction. So you actually end up with an overall increase in blood oxygen in that area. So even though the neurons are actually needing and taking more oxygen, you tend to get an overall increase in the blood oxygen. And that means that you have a different response in terms of the magnetic field relaxing back due to that, what we call a bold response, a bold oxygenation level dependent response. And by decoding the way in which the magnetic field is changed, you can then build up an image of the blood oxygenation. And by subtracting that blood oxygenation in the different parts of the brain from 2 conditions, you can find out which parts of the brain are more active in one condition than the other condition. So if I said, read these nonsense words and then read these real words, when you see the real words, you will be reading. In both conditions, you're still looking at letters, you're visually scanning the page, but only in one condition are you actually decoding. If I looked at the blood oxygenation dependent signal from those 2 conditions and subtracted the nonsense words from the real words, then I'd end up with the neural correlates of reading, those parts of the brain which are activating only because you are reading. And I'm touching this part of the brain, but there'll probably be more on that side, on the left hemisphere, actually. Okay, so that's fMRI. The spatial resolution in terms of visual detail is very good. So you can go into like 2 or 3 millimeters. The temporal resolution is okay. It's in terms of seconds. And some people, if they're doing EEG, would say, well, that's not okay. That's absolutely rubbish, because EEG is so sensitive in terms of time. But it depends what you're trying to do. The comfort of the participants is not that great, because you've got about 120 decibels of noise. You're inside a very confined tube. You're having to lie down, putting your head very often in a secondary magnet to improve the magnetic field measurement around the brain. So it feels a little bit like a, a sort of torture instrument or something going in there. It's becoming more comfortable. I mean, there was a time when you used to have to grip with your teeth onto a dental mold that had been molded exactly to your teeth so as to keep your head absolutely still. Now we have software that automatically adjusts. So in fact, we just track the movement of the head and we compensate and pre-process the data to remove those artifacts. So this is a picture of a very famous person. I'll just stand like that. You can probably recognize the shape of the nose. So you can do anatomical images with it, and you can also do functional images like that. So in actual fact, you actually pulse in a different way, and you grab a different type of response from the pulsed magnetic field in order to look at anatomy or to look at function. Slightly different sequence involved. And apparently there is no health limit to these, none that we know of. The actual biggest risk, do people know what the biggest risk is in terms of fMRI, in terms of health?
Like a magnetic metal
Yeah, metal. Anything metal. We really have to be careful of that. So when you participate in our experiments, in fact, I'm more careful in many respects than other researchers because I screen you twice. First of all, when you come along and say that you want to do it, and then you get screened again before you go into the scanner to make sure that you haven't got any metal in your body. So we ask you all sorts of strange questions like, have you ever been a metal worker? Have you ever been involved in an explosion? Even contraceptive coils, other things, all of those sorts of things. An awful lot of them actually we ask you about, not because they're particularly dangerous, but because they actually disturb the image that we create. But some of them could be quite dangerous as well. So we have to be absolutely careful that there's no chance of you taking metal in. If you go on Google, I'm sure you can find pictures of where metal chairs have accidentally found their way into the MRI scanning room, and they are just crushed completely flat against the magnet. You know, if you get between a piece of metal that's moving and the magnet, it will just pass through. It's pretty nasty. So we have to be completely careful of that. That's the only safety thing. But there's standard procedures for getting around those problems. And the safety record with them is excellent, and you can have as many scans as you like, as far as we're aware. Children are an issue because in neuroscience and education, we're increasingly thinking about putting children in these things. Children already do participate in these experiments. I haven't involved children yet in my studies, but it's something I probably will do in the future. I did put, actually, no, I'll be careful, I'm on camera now. I did put my child in there once, I have to say. He wasn't that happy. He was okay for about half an hour. It was when he was 9, it's my eldest. And then he said, actually, Dad, I think I've had enough now. I want to come out. And it just made me think, well, he feels he can say, I've had enough now, Dad, I want to come out. If I wasn't his dad, maybe he wouldn't have said that. Maybe he'd still be in there going, oh, I hate this. I'm not going to. So it did make me think about it. We have a wonderful scanning facility up the hill, which has just started. It's a 3 Tesla unit. And as I say, we will be having probably studies that you'll be able to participate in and volunteer to have a go in, if you like. And you get a free picture of your brain, which is really nice, in 3 dimensions, so you can sort of travel through it. You can print out pictures for Facebook and all sorts of other things as well. Okay, so that was most of what I wanted to say today. I've gone a little bit fast, so we're going to be ending probably a little bit before time. But it has given 15 minutes or so for questions, a bit of discussion. Because we have travelled so fast, I think it's important that we do that. Are there things that you would like to ask me? These are the things that we've talked about. Now remember, you're going to have a little exam on these, so it's good to be discussing them now. Yes?
You said before that if you have metal, you can go through the MRI. So what happens in cases...
Yes, you can't go through the MRI if you do have metal. Yeah, just to clarify.
What happens in the case, like, if someone has a cochlear implant? What happens then? Yeah,
I suspect. Well, there are websites where you can actually look at whether tests have been done on these individual devices. So I imagine it's the same with cochlear implants. So when people say they're wearing a coil, I have to get the code, the actual type that it is, and you go to a website and they say whether it's been tested with the magnet of that strength. So I don't know, but my guess is that some of the cochlear implants will be okay and some won't, and you just have to be absolutely sure beforehand. Yes.
Back to the myelin sheaths on the axon.
Yes.
Do we know why they're organised in the little bumps within the nose of ganglia in between? Wouldn't it be more efficient to just have it going all the way across? Ah,
the nodes of Ranvier. Okay. What function does the nodes of Ranvier actually? I'm not really sure about this, but we do have somewhere in the room graduates of neuroscience, yes, who might be there to what is the, because I mean part of it is because the myelin is being created by Schwann cells or oligodendrocytes, I imagine. So that's part of the reason. I'll just show that I know that, but I don't really know why we bother to have nodes of Ranvier.
It's actually faster, like the electrical whatever down the AP down in your by self-choice reduction. Like
it jumps between. So it's actually a lot faster on myelinated neurons. Like if you just had the whole thing myelinated, it wouldn't be able to jump from like, I mean, it can really travel down, but like it jumps between and it like makes it quicker.
Why couldn't you have a completely myelinated axon then, just jumps from one end to the next?
I just don't think it would work.
No, I
didn't know.
I mean, it may be because you need to have a system that you can actually mend, because these things are created by these cells traveling around. And I guess if it was a whole thing like that and it went wrong, then it would be.
The current has to flow through the axon, but when myelin's there, it can't do that. So obviously, like if say like the things switch around, like it's polarized on one end, then it wouldn't be able to travel down because it's too long a distance. Like you need that like gap where the current can switch over.
Oh, okay. Well, you'll be right. You'll be pleased to know that that question would not be in your multiple choice. Okay. Any other questions?
I suppose moving on from that, the cells that make the myelin.
Yeah.
Why do some of them make it across numerous cells, and some of them make it for a single cell? Is there single cells that only get it made by single ones?
Well, it may be to do with the fact that Schwann cells, so they only do single axons, and they work more in the peripheral nervous system, where there are much longer axons, whereas the oligodendrocytes work in the central nervous system, where there's much smaller. But this is almost like an evolutionary question in a way. Why is it the most efficient system? And I'm not. It's a difficult question to ask, but probably because of the different purposes or the actual dimensions of axons in peripheral and central nervous system. Unless anything to add.
Schwann cells and oligodendrocytes have really different structures. Like the cell bodies of Schwann cells actually wrap around the axon, whereas in oligodendrocytes, the cells like in the middle of it and it branches out. So I think it's just because in the brain all the neurons really clustered around each other, like you said, whereas in the peripheral, it's like one long nerve, so it's easier to wrap around the nerve. Whereas in oligodendrocytes, you've got a cell body in the middle and it's just easier to branch out because they're all clustered together. So we're more worried about that one. Yeah.
Yeah, we're not so worried about Schwann cells. It's nice to be able to say peripheral nervous system, we don't care. Okay, any other questions? Because I'm going to be asking you about all these things. So if anyone, it's a good time to clarify. Yes.
Take these as bullet points for revision for the exam. That'd be sensible.
I would look definitely at the slides for the exam, definitely. We will be doing a sort of mock in Teams at some point using Zondel Team Play, which is like a sort of. So we will be having a little experience of the sorts of questions that you will receive, but definitely I would look at the slides, the two sets of slides. Yes.
Is it going to be something specific as kind of what does this do?
Yes, there will be an explanation. Well, there might be. I'm not saying there will be. There might be an explanation of how an action potential occurs in terms of polarization and depolarization with words missing and that sort of thing. There might be a picture of a neuron saying, "Can you please label this?" Whatever. But they are multiple choice, so you have to choose the things. Yes. Oh no! Oh no! No! Absolutely not. Do not learn this, please. Okay. Sorry. This. I'll tell you the reason. The reason I put this was because every year people will say to me, "This plastic brain is very interesting. What is number 35?" And I go, "Oh, I'm not sure, but it's on here." Okay. That's why I put it there. I mean, I think. It may well be handy in a minute if you want to stay, because we've got a little bit of time. And if you want to stay behind, you want to carry on looking at this brain in relation to that piece of paper that tells you the different parts, please by all means do. But any other questions about, looking back? Some of the things that we've talked about. So you might get a picture like that and you might be asked to label it. There might be an explanation with words missing. That might be on there as well. Yeah, please. I'm just using this to stimulate questions. Yes.
Could you maybe just go back to the other slide?
Absolutely.
One with the network of like 16 neurons you have.
Yeah.
And I didn't quite understand how a message can change.
Oh, it's a very good question, because I didn't explain it terribly well, because next week we're going to be looking at learning. But I will say a little bit more about that, because what all you need for the information here to become different type of information here is for there to be differences in the efficiency with which the electricity is actually passed on. So if, for example, it's a very difficult thing to explain this because it's mathematical, and actually it's kind of difficult almost to believe. You have to see one of these networks operate. But essentially, if you have these connections here, so that the efficiency with which this electricity is passed across is different here to the way it is there, and you fix those efficiencies, then you can actually make the output here respond to anything you want in relation to this. So in an artificial neural network where they've actually simulated this with a computer, what they do is they put on a 1 here and a 0 there, and they go, no, 0. A 0 there and a 1 there, and they go, no, there. And it back propagates and actually changes the efficiencies of these so that it exactly maps whatever you put here to whatever you want to see there. And then after a while, once you've got the right weights, you can produce anything here you want in relation to what you put in there. So, that's you're beginning to think, I'm beginning to talk about how you learn with this. So you can think with it, because information in is something different to information out. But the learning comes when you actually change these efficiencies in a particularly helpful way, So, yeah.
I was just wondering, on the very second slide, you said how communication works within neural networks, and you mentioned there are 2 emissions, AP. And,
was it this slide?
The second.
Here. How communications occurs within them?
Yeah.
Yeah, so that stands for action potential. That's action potential. That's the electricity passing down the axon. Oop.
Paul, I have
Yeah.
a question. With the action potentials, the depolarization needs to reach a threshold, yeah?
Yeah.
What instigates that threshold? What starts it all going?
What starts it all going? Well, you have to have something that starts it all going, and that's the input to the body. And you have various inputs to the body. And these are usually peripheral neurons. So they could be like a nociceptor, it could be like a touch peripheral neuron that actually, when you press it, it fires, if you like, or touch it, it fires. It could be a sensory neuron, like a visual peripheral neuron that actually, when it receives light, it fires. So these are special type of neurons that are at the periphery, and they correspond to the absolute input of the human body. I should just run this through these. I'm not going to keep you much longer if you're in a hurry to get off. But just reminding you of what we've done. You're okay with these terms? Okay. And those terms as well, those are definitely worth learning. You do not have to learn this. Important. Do not learn this. This was really just for information in terms of forebrain, midbrain, and hindbrain. I think that's important to know. You need to know where the corpus callosum is, where the cingulate cortex is. I'm not actually going to be asking anything about cephalons, I don't think. You need to know what the medulla, the pons, the cerebellum is about, and roughly where it is. By the way, you can make these. There's a website, apparently, I've found that you can actually make these up, these images. Excellent. You need to know about the tegmentum and the tectum. I don't think, actually, to be fair, I don't think you need to know too much about the small print here. All right, this small print you don't need to know about. Okay, I don't want you bothering to learn that at the moment. Just know that the tegmentum is this area here, and the tectum is that area there. You need to know where the thalamus and the hypothalamus is and what it does. Also need to know those areas, the thalamus, the striatum, nucleus accumbens, and amygdala. I found it actually very helpful going on Google and looking at all the different images of the same thing. Because you look at it at all different angles and different attempts to try to make it clear, because it's not very easy seeing these three-dimensional things. And actually, seeing lots of different ways of representing it, I found very helpful. Although I do like these rotating brains. You need to know a gyrus and a sulcus. Definitely need to know your 4 lobes. They should be just tripping off your tongue. Yeah, you need to know where those are. You need to be able to find different regions from their names. You do not need to learn all your Brodmann areas. Very important, please. Do not learn your Brodmann areas. I just need to know that BA, what they are, okay? But do not learn where Brodmann area 5, 7, and 8 are or anything like that. I think I would try to understand this diagram and understand where unimodal and multimodal association areas are. You need to know what the advantages are and disadvantages are of the different ways of studying functional neuroanatomy. Okay, so you need to know what's good about PET, what's bad about PET, what's good about fMRI, bad about fMRI, etc., and EEG. You need to understand the difference between alpha, beta, and theta. Yeah. You need to be able to explain functional magnetic resonance imaging and understand the pros and cons. That's about it, really. And then we're going to have next week's as well. Now, I realise that some of you are not going to be able to make next week's. Most of you, I think, can. Put your hand up if you can make Thursday at 9 o'clock. Actually, I'd better say, put your hand up if you can't make Thursday at 9 o'clock. Okay, so those people, I do apologise to for this. I'm going to try to, hopefully this has worked. Hopefully the quality is good enough, and I will try to put it on the net for those people. Okay, final questions. I hope you've enjoyed this lecture. You can take the paper bits away. What I would like you to do, though, and I really would appreciate this, is if you could just nominate one person. Oh, have you signed the register? There should have been a register going around. What's happened to the register? There was definitely a register. Oh, it's right at the back. Can you make sure that goes right? Can you nominate one person on each table, please, to put their brain together? When you finish looking at it, please stay for 10 minutes and look at it if you want. But when you finish with it, can you please put it in one of the little cardboard boxes and then put the little cardboard box in the big cardboard box in the back? Any questions? Thank you very much. I hope you've enjoyed the lecture. You should have received an email already telling you where to find it on YouTube. It will be there in about 5 or 10 minutes. So.